Hello everyone and welcome to our module on trinucleotide repeat disorders. Trinucleotide repeat disorders are genetic disorders that occur in genes with repeated trinucleotide units. So for example, some genes contain the nucleotide CAG repeated over and over again. This is the type of gene where one of these disorders can occur. Most of them involve the central nervous system in one manner or another, and we'll talk about all the examples listed on this slide in this video, including Fragile X syndrome, Friedreich's ataxia, Huntington's disease, and myotonic dystrophy. The genes involved in all of these disorders share some common features. For all of these genes, there is a wild type or normal allele that is found in most individuals. This allele is polymorphic, meaning that it has many forms, and those many forms include a variable number of repeats from person to person. However, overall, even though normal individuals may vary in the number of repeats found within their gene, the overall number of repeats is relatively low. In the disease version or abnormal version of the allele, this is what is found in affected individuals, and there is an increased or expanded number of repeats beyond the normal range, and this is what leads to disease. And the reason that these sections of DNA have varying numbers of trinucleotide repeats is probably related to slipped DNA mispairing. I talk about this in the video on DNA mutations, but this can occur in sections of our genome where there are repeated units, and DNA polymerase gets confused when copying the genome, and it makes fewer or additional copies of that trinucleotide repeat. In all of the trinucleotide repeat disorders, the disease gene demonstrates what's known as unstable repeat expansions. What this means is that in addition to the disease gene having more repeats than in normal individuals, the number of repeats may also increase in the offspring, such that from one generation to the next, there are more repeats in the disease gene. And a key point here is that this makes these trinucleotide repeat disorders unusual in that the genetic abnormality may change over time. We'd like to think of mutations as going along from one generation to the next and being the same mutation. That is not the case in these disorders. With each additional generation, you may see more repeats in the disease gene, and that's called unstable repeat expansions. Because of these unstable repeat expansions, a phenomenon may occur called anticipation. This is where the disease severity worsens in subsequent generations with earlier onset in subsequent generations and sometimes more severe disease. And this is because in additional generations there are more repeats accumulating in the abnormal gene. The first trinucleotide repeat disorder we'll discuss is Fragile X syndrome. This is an X-linked dominant disorder that results from an abnormal FMR1 gene. FMR1 stands for Fragile X mental retardation 1 gene. It is a gene found on the long arm of the X chromosome. Most commonly, the abnormality in the FMR1 gene is an increase in CGG repeats. Normal individuals have less than 55 repeats in their FMR1 gene. In individuals with the full mutation leading to Fragile X syndrome, they have greater than 200 repeats. And when there are this many repeats in the FMR1 gene, that leads to DNA methylation of the FMR1 gene. I talk about DNA methylation in the video in the biochemistry section on DNA structure. It is a mechanism by which genes can be silenced, and when the FMR1 gene becomes methylated, this leads to silencing of the gene, and therefore it doesn't function normally. Fragile X syndrome is more severe among males. Even though it is X dominant, because males have only one copy of the X chromosome, they have absence of a normal copy of the X chromosome, and that leads to a more severe syndrome. Fragile X is the second most common genetic cause of intellectual disability. Down syndrome is the most common. An easy way to remember the features of Fragile X syndrome is to just think of it as a Down syndrome variant. Children with the Fragile X syndrome can also have anxiety and ADHD. They often have features of autism. They have a characteristic facial appearance with a long, narrow face, large ears, and a large jaw. This is a picture of a child with Fragile X syndrome shown on the screen here. And then finally, a classic feature of Fragile X syndrome is large testicles. This is referred to as macroorchidism. The next trinucleotide repeat disorder we'll discuss is Friedreich's ataxia. This is a hereditary ataxia disorder, and I discuss the clinical symptoms in more detail in the neurology video on the cerebellum. Friedreich's ataxia is an autosomal recessive disorder. It's caused by a mutation of the frataxin gene on chromosome 9. This gene is necessary for normal mitochondrial function, and the mutation that occurs is an increased number of GAA repeats. This leads to decreased frataxin levels inside of cells. Because frataxin is a mitochondrial protein, there are high levels found in the brain, heart, and pancreas. These are organs that depend heavily on mitochondria. So when there are abnormal frataxin proteins, this leads to mitochondrial dysfunction, principally in these structures listed on the screen here. In the cerebellum video, I discussed the clinical features in more detail, but I'll briefly review them here. Friedreich's ataxia begins in adolescence with progressive symptoms. There is cerebellar and spinal cord degeneration. That's why this is called a hereditary ataxia. Patients often have loss of balance and weakness. This is associated with hypertrophic cardiomyopathy, and that's a high-yield detail that you should remember for the Step 1 exam. And in addition, patients typically develop physical deformities, including kyphoscoliosis and foot abnormalities. Our next trinucleotide repeat disorder is Huntington's disease. I discussed this in detail in a video in the neurology section, but I'll briefly review its features here. Huntington's disease is a movement disorder that involves the central nervous system. It's an autosomal dominant disorder that is caused by mutations in the HTT gene, which codes for the protein huntingtin. In patients who have the mutation, there is an increased number of CAG repeats, and because CAG codes for glutamine, this is often called a polyglutamine disorder. There are a couple of these other than Huntington's. The others are all very rare, but they are all central nervous system disorders that involve an increased number of CAG repeats. Just like in all of the trinucleotide repeat disorder genes, the HTT gene normally has 10 to 35 repeats found in normal individuals. In individuals with Huntington's disease, there are an increased number of repeats found from 36 to 120. The symptoms result from degeneration in the basal ganglia, specifically the striatum. This can lead to symptoms of chorea. There's also typically dementia. The onset of symptoms is usually in the 30s to 40s, and death usually occurs after 10 to 20 years. Our last trinucleotide repeat disorder is myotonic dystrophy. This is a muscle disorder. It's considered one of the muscular dystrophies, along with Duchenne and Becker muscular dystrophy, and myotonic dystrophy is an autosomal dominant disorder. There are a number of subtypes of myotonic dystrophy. The most common type is called type 1. This is caused by an abnormal DMPK gene on chromosome 19. DMPK stands for dystrophia myotonica protein kinase. The abnormality in the gene is usually a CTG repeat expansion, and this gene codes for a protein called myotonic dystrophy protein kinase. When the CTG expansion is excessive, this leads to an abnormal gene that gets transcribed into MRA but never gets translated, and therefore there is loss of the myotonic dystrophy protein kinase. The less common type is called type 2. This involves a different gene called CNBP. This is much more rare. It's usually a less severe disorder than type 1, and interestingly, this is a CCTG or tetranucleotide repeat disorder. It is not a trinucleotide repeat disorder. In the more common type 1 form of myotonic dystrophy, symptoms begin in adulthood, usually in the 20s or 30s. This is much later than the symptoms that begin in other muscular dystrophies like Duchenne and Becker. There is progressive muscle wasting and weakness, and the reason it is called myotonic is because patients usually exhibit myotonia, which is prolonged muscle contractions. They're often unable to relax their muscles after use. For example, classic symptoms are inability to release the grip or locking of the jaw. The facial muscles are often affected, and this gives patients a characteristic facial appearance caused by muscle weakness and wasting. This is a picture of a man with myotonic dystrophy shown on the screen here. Patients typically have a long and narrowed face and hollowed cheeks. And one thing that's important to remember about myotonic dystrophy is that it's a multi-system disorder that involves many non-muscle systems of the body. Patients with this disorder typically display hypogonadism, cataracts, cardiac arrhythmia, and they often develop frontal balding, just like this man shown in the picture here. The hypogonadism is a primary hypogonadism. The testes release low levels of testosterone. The FSH level is usually elevated. These patients typically have oligospermia and infertility, and they can develop testicular atrophy. In addition, other endocrine systems can be involved, and insulin resistance in particular is common. The heart can be involved in myotonic dystrophy just like it can be involved in other forms of muscular dystrophy. However, in myotonic dystrophy, it's more often arrhythmias that develop as opposed to heart failure. Patients can develop a first-degree AV block. This is the most common finding. They can also develop bundle branch blocks. They also often develop arrhythmias like atrial flutter and atrial fibrillation. There's a high prevalence of cataracts in patients with myotonic dystrophy. Also, they occur at a younger age, and for this reason, these patients are often referred for regular slit lamp exams to screen their eyes for early development of cataracts. The lungs are often involved, and respiratory complications are common. This usually results from weakness and myotonia of the respiratory muscles. This leads to a decreased vital capacity, and alveolar hypoventilation, and respiratory failure can occur and can be a mode of death. And then finally, intellectual disability is common in myotonic dystrophy. The severity is worse with a younger age of onset. So for example, individuals who develop myotonic dystrophy in childhood often have severe cognitive impairment throughout their lives. And that concludes our video on trinucleotide repeat disorders.